Hey guys, so today we're teaching you about DNA replication, and this is an original video by Miss Rosales' third period class starring myself, Leah Alexander, Mario Sanchez, Alejandro Hernandez, Xenia Lara, and Brianna Torres.
Hey guys, now starting with the comparison of DNA to RNA. In DNA, it is a double-stranded and runs anti-parallel, which means it starts with the 5' and ends with 3', and vice versa. In DNA, adenine binds to thymine and cytosine binds to guanine by hydrogen bonds. These bonds are so strong that they do not actually touch, and you'll see why. In DNA, there is a sugar phosphate backbone, and the sugar in DNA is deoxyribose. In DNA, adenine binds to thymine, which is a purine binding to a pyrimidine. In RNA, it is single-stranded. Adenine does not bind to thymine, it binds to uracil, and guanine binds to cytosine. There are 3 types of RNA: messenger RNA or mRNA, rRNA, which is ribosomal RNA, and tRNA, which brings AA to the ribosome. There's also ribose, that is the sugar, and then uracil is the pyrimidine in this case.
Structures of DNA. Phosphate. Hold on. Pause. In the structure of DNA, phosphates, sugars, and the nitrogen bonds bind together, and the nitrogen bonds are adenine, thymine, guanine, and cytosine. And the main sugar is deoxyribose. In anti-parallel, 5' runs down with 3', and then 3' runs down to 5', and the hydrogen bonds hold the nitrogen bases together.
Okay guys, back to DNA. In genetic coding, it can be used to trace common ancestors and study the evolution of organisms. Now on to background and history of DNA. The history of DNA really starts off with a series of experiments and discoveries. Rosalind Franklin was the first, she was the first woman to really be known. She is very known for discovering that DNA has 2 strands and nitrogen bases are inside the DNA helicase, and that sugar phosphate is actually outside instead of the inside. In the Hershey and Chase experiment, it is a series of conducted experiments to prove that bacteria could be transferred from one strain to another, and that is in 1952. In 1944, Avery, McLeod, and McCartney found that the S strain cells to prove that the DNA was responsible for the transformation of the bacterial cell. Watson, Crick, and Wilkins. These 3. So basically, they stole all of the other scientists' ideas that were to determine sugar and phosphate were on the outside, determine that nitrogen bases were on the inside, just like Rosalind had said. And Wilkins was Franklin's boyfriend, and he basically delivered all of her data and information that she had discovered to Watson and Crick, and they basically all got credit for someone else's work.
Leading and lagging. 3' to 5' is leading strands, 5 to 3' is the lagging strands. The orientation of primers means the direction in which the elongation of the primer in DNA synthesis occurs. It is also read from 5' to 3' direction. New nucleotides can only be added to the 3' end.
Okay, now on to DNA replication process. In the DNA replication process, you have a helicase, which is a protein that unwinds the double helix, a primase that acts as an enzyme responsible for adding the primer to start DNA replication, DNA polymerase, which adds new nucleotides to the 3' end of the strand, ligase, which joins Okazaki fragments together. Okazaki fragments, by the way, are fragments that are made from the replication of lagging strands. You have leading strand, which is 3' to 5', and lagging strand, which is 5' to 3'.
Errors in meiosis and mitosis. Trisomy 21 when there is an extra chromosome at the 21st pair, also known as Down syndrome. Karyotype. A picture of an individual chromosomes goes from biggest to smallest pairings. Last are the sex chromosomes or gametes. An euploidy, the presence of an abnormal number of chromosomes. Some examples are 45 or 47. Turner syndrome. Chromosomal disorders in which a female is born with only one X chromosome, and that is a sex-linked trait. Chromosomes, numbers, and structures. They can change during the duplication of cells during anaphase if there is a wrong chromosome split, or the structure can change in the same spot.
The components of a nucleotide. There are 3 main components of a DNA nucleotide. A nucleotide is composed of a sugar, which is also known as deoxyribose for DNA. One of the 4 nitrogen bases, which include adenine, thymine, guanine, or cytosine. A phosphate group, which is 1 phosphate joined by 4 oxygen atoms, just as in the illustration below.
Purines versus pyrimidine. Purine is double ring, which is adenine and guanine, 2 carbon containing nitrogen ring bases. Pyridine is a single ring, which is thymine and cytosine, 1 carbon nitrogen ring bases. These bases form hydrogen bonds with their complementary pyrimidines, also known as complementary base pairing.
You can also remember this as angels and gods are pure, so they have 2 rings. This is a little trick that our bio teacher has taught us to remember the difference between purine and pyrimidine. Base pairing mutagenesis. Mutagenesis is a process by which mutagenesis is when genetic info is changed, leading in a mutation. These changes can happen abrupt and heritable, induced by mutagens, which are also chemical or physical agents. In base pairing, you have the normal base pairing, which substitution can occur and the base pair will be changed, but no mutation will occur because you have the same amount of base pairs that you started with. But if deletion occurs, then you will not have enough, and all the bases will run to where they are looking for a pair, but they cannot find one, causing a mutation in the gene. In insertion, you would have too many, and it would not have enough because you need 3 pairs to complete the base pair. So that is why substitution would not change the base pairing in mutagenesis, but deletion or insertion would.
Okay guys, thank you so much for watching. If you like this video, go ahead and check out all of our other videos. The link will be in the description. That's all. Thank you.